Hi, hope you're doing well. So, this is biology HL paper 2 and time zone 2. So, the first question is mammalian tissue are circulating nutrients, including glucose, amino acids, and various intermediates, metabolites for respiration. So the concentration in the blood of glucose and other metabolites is regulated as a part of homeostasis. Okay, the concentration of metabolites remain constant if it is absorbed from arterial blood and consumed by metabolism at the same rate. Okay, circulatory turnover flux is the rate at which metabolite wait so yeah so metabolite in the blood is both consumed and produced, with the concentration in the blood remain constant. So circulatory turnover flux were measured in fasting mice that had not fed for 8 hours. The table shows data for metabolites that had the highest circulatory turnover fluxes. Alanine, glutamine, glycine are the amino acids. So circulatory turnover flux values are shown per grams of mouse body mass. State one advantage of this. So this turnover basically make a comparison between the mouse and the other animals. So that's the advantage. That's the function of the circulatory turnover flux. We can compare the different organisms by this kind of circulatory fluxes. So comparisons between mice and other animals. That's the answer. Okay, identify the metabolite with the highest circulatory turnover flux and the amino acids with the highest blood circulation. So you can say the highest is 374.4 and yeah, that's the answer. And the mean blood, so the highest circulatory turnover is lactate, amino acid with highest blood concentration. So you have to check the blood concentration with respect to circulatory turnover. When we have very small difference. So that difference is the smallest. So that's why the amino acid with the highest, like when we calculate the concentration, so it should be glutamine. We can calculate it, that is glutamine. Deduce, giving a reason for your answer, which type of molecule is likely to remain in circulation for the longest before being absorbed by the cell, which has the lowest turnover flux. as compared to mean blood circulation, palmitic acid has the lowest circulation, we can say that, turnover, circulatory turnover flux. So the answer is palmitic acid, because it has lowest turnover flux with respect to blood concentration. That is the answer. Next question, circulatory turnover flux were also measured in mice that had been feeding instead of fasting. The graph shows the result for glucose, lactate, glutamine in both fasting and feeding mice. So mean results are indicated by horizontal lines on the graph and circulatory turnover flux for each mouse by an individual data point. So outline the changes to circulatory turnover fluxes that would occur in a fasting mouse that started feeding. So you can see, you can easily see the circulating turnover flux increases for feeding than fasting. So feeding then fasting. So the first point we can say that, like, flux, it would be increase for feeding then fasting. That's the one thing we should know. And there is the significant increase for glucose. Look at this. That's the significant increase. So the most significant increase for glucose. And for lactate, the percentage is very small. So smallest percentage increase in lactate. That's the answer. So using your understanding of insulin secretion, explain the change in glucose circulatory turnover flux between feeding and fasting mice. So yeah, so insulin secretion. So insulin, it basically controls the blood glucose. So whenever the blood rise in blood glucose, when like glucose produced by digestion and absorbed in small intestine and insulin secrete in feeding mice. So Yeah, we can say that. Like insulin secreted in feeding mice, and insulin causes more metabolism of glucose because it lowers down the blood glucose level. So insulin cause uptake of glucose into glycogen because glucose stored in the glycogen form. So we can say that this flux increases because glucose metabolizes faster, since glucose metabolize faster. That's the answer. Okay. So the group of fasting and feeding mice were infused with radioactivity labeled lactate, glucose or glutamine. Radioactive labeling of Krebs cycle intermediates was then monitored to determine the relative quantities of three metabolites that were being absorbed from the blood and used in the Krebs cycle. So the bar charts show the contribution of each metabolite to carbon entering the Krebs cycle as the proportion of the total in different parts of the body. Okay. Identify the organ that metabolize the most lactate in both feeding and fasting mice. Most lactate. So look at the kidney. So answer is kidney. The brain is unlike other organs in its use of metabolites for Krebs cycle. Distinguish between the data for the brain and all other organs of tissues. Glucose, brain need 24/7 the glucose. 24/7. That's why. So we can say that brain uses more glucose yeah and also like brain use more total proportion of these metabolites yeah That's the answer. So using any of the data in question 1, evaluate the hypothesis that the main metabolite absorbed by cells and used in respiration of glucose. So yeah, only for brain. The other things, the other one, they are not like supported this hypothesis. So that's why we can say that like hypothesis is not supported except for brain. And for the lactate turnover, highest turnover for the lactate as compared to glucose, you can see. Yeah, you can see in both fasting and feeding, the lactate is higher than glucose. Than glucose. That's the answer. Okay, question number 2. The structure of the transfer RNA molecule is shown in the diagram. The attached amino acid, which is aspartic acid, is indicated by arrow. Reduce the function of CUG. That is anticodon. Explain how the living organisms ensure that amino acids linked to this transfer RNA molecule always aspartic acid. Enzymes always ensure that specific amino acids binds to transfer RNA. Enzyme always make sure that specific amino acid binds to transfer RNA. And this transfer RNA activating the enzyme. It activate enzyme. And enzyme binds to transfer RNA and recognize the shape of transfer RNA. And different activating enzyme for different transfer RNA. There are like different activating enzyme for different transfer RNA. And then like attached amino acid to anticodon. That's how you'll get the marks. During translation, 3 binding sites of transfer RNA molecule are used. Outline how each of these binding site is used. So that's the process of translation we have to do. Like binding sites are on ribosomes. Are on ribosomes, a site for attachment of transfer RNA. Yeah, so this is the site for attachment of transfer RNA and where the peptide bond is made. Okay, state the molecule that trees such as redwood use as a genetic material. So that is very easy. They use DNA. Okay, if the genome of the 5 new trees were sequenced, predict with a reason how similar they would be to each other. So yeah, they are identical. They are of course identical. Why? Because that is cloning, like asexual reproduction. Because of asexual reproduction. That's the reason. And the clone produced by cloning. And outline one method that has been successfully used for the cloning. Yeah, you have to explain the cloning process. First of all, like nucleus removed from egg. That's the first step. Then body cells removed from donor. Then we have to enucleated egg and body cells, and then embryo implanted. Embryo implanted. That's how you get the 3 marks. Okay. Then like Bateson and Punnett carried out a series of genetic cross using variety of sweet pea. So they cross 2 varieties. Okay, state the genotype of the F1 hybrid. So they should be capital capital C, small c, capital R, small r, because F1 hybrid, yeah, that is the answer. Explain how the pattern of inheritance result in 9:7 in the F2 generation. So we have to cross. We have to make the cross. We have like capital C, small r, capital C, capital R, capital C, small r, small c, capital R, small c, small r. Capital C. So we have capital C, capital C, capital R, capital R, capital C, capital R, small c, capital C, R, then capital C, small c, capital R, small r, capital C, this, 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 this. So that's the result. So we have purple, so purple, 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 we have purple, 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 purple. So these are, we have purple, purple. Okay, so you can easily see. So this is our F2 generation. So we clearly see this is 9:3:3:1. So which is, if phenotypically we see this is 7. So 9:7. 7 is white and 9 is purple. So that's how you will get the 3 marks. Okay, predict the outcomes of crossing plants with the genotype capital C, small c, R. Yeah, so this is very easy. We will have, that's a very easy cross. Can make the cross. Capital C, small r, small c, small c, capital R, small c. So we have capital C, small c, capital R, small r, small c, capital R, small r, capital C, small c, R, R. Then we have small c, small c, small. So we have one purple, three white. One purple, three white. Okay, plants have cell wall composed of cellulose. Describe the structure of cellulose molecule. So cellulose is beta glucose. It is made up of beta glucose in which OH group in the beta position. So the binding is like C1 and C4 bonds of C1 and C4 glycosidic. So we can say that this is 1,4 glycosidic linkage. 1,4 glycosidic linkage is formed. This is unbranched, glucose is, cellulose is unbranched and it is straight chain. It is not spiral. And glucose subunit, they oriented alternately upward and downward. Upward and downward. So hydrostatic pressure was measured in onion epidermis cells. Okay, outlines how pressure higher than atmospheric pressure develop inside plant cells. So basically, when water is entered by osmosis, since the solute concentration is higher inside, so water potential is more negative. So that's the reason. So water enters by osmosis, and we can say that like solute concentration is higher inside the cell, so water is more negative inside. And since the cell wall does not expand, so cell wall prevents the cell from increasing in volume. That's the answer. State the importance of cellulose to the plant when pressure is higher inside than outside. So it prevents bursting because cell wall is very, very having the compact structure. So it prevents bursting. When a plant cell grows, cell wall must expand. Explain the role of oxygen in cell wall expansion. Oxygen causes specific genes to transcribe. Transcribed. So it stimulates the proton pumps. Proton pumps cause movement of hydrogen ions into cell. And hydrogen ions make the low pH, which make pH low. And it breaks connection. It breaks the connection between cellulose. That's the answer. Yeah. So next is okay, next is answer 2 questions up to additional mark available. Okay. So excretion is one of the function of life. Right. Outline how carbon dioxide carried out to lungs into pulmonary artery is moved to air out of the body. Yes, easy question. So 6A. The carbon dioxide, excreted by alveoli. Alveoli. So capillaries surrounds the alveoli wall and diffusion of carbon dioxide occurs, and carbon dioxide move to blood, and then once the carbon dioxide moves to blood, from blood to air, carbon dioxide move from blood to lungs, and then by diffusion and high concentration of carbon dioxide in blood. So then by ventilation it move outwards. By ventilation it move outwards. So you have to use the words in marking for this question: ventilation and airflow of lungs. This is very important. Then you will get the marks. And also you have to use the word for this question: contraction of muscles, because contraction of muscles is very important. Muscles of abdomen muscles is very important. This is how you will get the marks. So that's a very easy question. You have to explain this question with respect to like abdomen muscles contraction, then ventilation, flow of carbon dioxide, that's by diffusion, and like the capillaries network which actually receive the carbon dioxide, it has the short distance, so it can easily pass the carbon dioxide to the alveoli. And that's how you have to explain this process. Now the question number B. Explain how the kidney produce the urine with higher concentration of urea than blood plasma. That's also very easy question. You have to explain. Ultrafiltration is the process. Urea is the thing which we won't need anymore. Rest of the things like they reabsorb. So ultrafiltration is the process in Bowman's capsule, in which like after that water reabsorbed, and we have like proximal convoluted tubule, that is basically the collecting duct. It is the place where the reabsorption occur. Solutes, ions, amino acids, these are the things which actually reabsorb by that duct, PCT duct. Reabsorption occur. So, but urea is the thing, there is no channel for urea, so no urea reabsorption occur. We have the hormone ADH, antidiuretic hormone, it causes more water reabsorption. Water reabsorption occur, but urea, there is no channel for urea reabsorption. That's how urea is formed, and urea is excreted out. That's how you will answer this. Compare and contrast excretion in humans and unicellular organisms such as paramecium. So both produce carbon dioxide. Both produce carbon dioxide. That's the similarity. And the difference is paramecium does not have specialized organs. It doesn't have special organs for excretion. and also paramecium excrete through plasma membrane. By passive diffusion. That's the difference. And the last thing which is very important, human, they excrete urea, whereas paramecium, they excrete ammonia. That's the end of the 6th question. Now we have to do 7. Describe how population in communities rely on each other for the supply of energy. So yeah, that's question 7a. So like the autotrophs, they provide energy. Autotrophs, they provide energy because that's the prime source of energy. Then autotroph also convert light energy into chemical, they convert into chemical energy by photosynthesis, and then that chemical energy, they flow through food chain, they move into food chains, and then in food chain there are some heterotrophs which rely on carbon compounds from the other organisms. They consume and they are consumers which ingest the food, and then there are like, we have to explain between primary consumer, secondary consumer. Primary consumer get the food directly from the autotrophs, then secondary consumer get from the primary consumer. That's how like food chain. And then there are saprotrophs. They are basically decomposers. They get the food by decompose the material, feed on dead organic. So yeah, this is the thing. You have to explain the entire process for 7 mark. I explain how plant cells organized, originated by endosymbiosis. So the large cells, like when there is the host cell with nucleus, they engulfed by bacteria. Then after that, endocytosis takes place. So in endocytosis, then like we can say that the aerobically respiring bacteria, they have there is aerobic respiration in bacteria, because there are certain bacteria which have the aerobic respiration material, and then that's how the mitochondria evolve. That's the evolution of mitochondria. evolution. And then from mitochondria, and then in the same way, there is a bacteria which is called cyanobacteria. So cyanobacteria, they are photosynthetic. They turn into chloroplast. That's how they are formed. You have to explain this process of, this is called endosymbiosis. They all have the DNA and ribosome of 70S, smaller. Analyze the relationship between plants and their pollinators. So basically, there is a mutual relationship between them. So the pollinators, they transfer pollen from anther to stigma, and then transfer pollen between plants, and then then we can say that pollens transfer to convert into nectar, then plant gains higher chance of pollination because pollens to stigma, and then after that there's fertilization occurs. So that's the whole process you have to explain. Like what happens? Like pollinator, they transfer pollens from anther, then from anther to stigma, and then transfer pollens between plants, and then plants, they gains higher chance of pollination. That's how you will explain this. So that's why we can say that that is the relationship between plants and their pollinators. Like this in 7C, you have to write that's a mutual relationship. Pollinator transfer pollens, and these pollens, you can say that it transfer from plant to plant, and these transfer of pollens between pollen and this this process actually increase diversity. You also have to mention this point as well. This portion increase diversity. I hope you understand this. So you have to attempt only 2 questions, but still I'm going to explain this as well. Species are groups. Okay, outline how new traits in species can develop over the time. This is the question number 8. So mutation actually generate the variation. They always generate variations. So how? Because mutation, they change base sequence. So when the base sequence is going to change, what happens? New alleles are going to form. When new alleles are going to form, then like better adaptation occur, and that's how, and these genes, they pass from generation to generation. Gene passed on. And this is like, that's how the directional selection caused the change in trait. These directional selection causes change in trait. And then another very important thing, which is like antibiotic resistance occurs. Antibiotic resistant traits form. That's very important. So that's how the developing new traits over the time is that's how evolution occurs. Then like explain how a new species can form abruptly and by polyploidy. You know, polyploidy is having more than 2 sets of homologous chromosomes. Whenever you see a term polyploidy, that means more than 2 sets of homologous chromosome, and triploid having 3 sets. Triploid having 3 sets of homologous chromosome. So polyploidy cells, they generate from diploid cells by DNA replication without cytokinesis. So whenever there is a DNA replication, during DNA replication, without cytokinesis occurs, this form polyploidy. And this fusion of diploid and haploid gametes result in triploid. Fusion of diploid and haploid gametes make triploid. So diploids and tetraploids are reproductively isolated. diploids, and tetraploids. Tetra mean 4. So di and tetra are reproductively isolated. Diploid and tetraploid are reproductively isolated, and the polyploid are therefore different. They are new species. The polyploid, they are like, they have different or new species. And advantage of some of system for naming species that scientists use. So you know the binomial system, in binomial system there are two names. The first one is genus, the second name is species. So internationally agreed system, that is agreed system, and genus indicates the most closely related species. So that's why we can say that advantages is like it is internationally accepted. Internationally accepted system. So genus indicates the most, this genus indicate most closely related species, and species, it is like, you know, it indicates the species. So it's easier to name newly discovered species. Easier to name newly discovered species. That's it. So thank you so much.
Thank you for staying with me to the end. Don't hesitate to leave your question or request in the comment section. I'll see you guys next time.